Hello everyone, welcome to my YouTube channel. I hope you all are fine. So, welcome to another video of BRS Physiology lecture series. So far, we have completed the action potential and resting membrane potential. I hope everything is clear to you till now. And in this video, we will be talking about the neuromuscular junction. But before that, I want to talk about the chemical synapses. So, let's look into this diagram. Okay, so synapse, it is a point of communication between two neurons, like in this diagram, or between a neuron and a target cell, like muscle or gland. And at the synapse, the firing of an action potential in one neuron, the presynaptic or sending neuron, it causes the transmission of a signal to another neuron, that is the postsynaptic neuron or the receiving neuron, making the postsynaptic neuron either more or less likely to fire its own action potential. Okay, so in the second diagram right here, see that we have a presynaptic cell and a postsynaptic cell. In the presynaptic cell, there are many synaptic vesicles. These are membrane bounded spheres filled with neurotransmitter molecules. Also, see that there is a small gap between the axon terminals of the presynaptic neuron and the membrane of the postsynaptic cell, and we call this gap as the synaptic cleft. And the membrane of the postsynaptic cell, it has receptors for the neurotransmitter. And the third diagram is kind of self-explanatory. See that the action potential, it reaches the axon terminal and it causes the depolarization of the membrane. And in response to this depolarization, we have the depolarization sensitive calcium channels. And these calcium channels open, and we know that the calcium is present in the higher concentration outside the neuron than the inside. So this calcium rushes into the cell, and the calcium allows the synaptic vesicles, which contain the neurotransmitter, they fuse with the axon terminal membrane, and thus releasing the neurotransmitter into the synaptic cleft. And this neurotransmitter, it binds to the receptors on the target cell. Okay, now open your BRS physiology book and we will get through this together. The first thing we have is the general characteristic of the chemical synapses. Uh, the first point is the action potential in the presynaptic cell. It causes the depolarization of the presynaptic terminal. So when the action potential it reaches the presynaptic cell, it causes the depolarization of the presynaptic terminal, and as a result of depolarization, calcium enters the presynaptic terminal through the calcium channels, the depolarization sensitive calcium channels that I have talked about in the diagram, and this causes the release of neurotransmitter into the synaptic cleft. And the neurotransmitter, it diffuses across the synaptic cleft, and then it combines with the receptors on the postsynaptic cell membrane, and this causes a change in the permeability to ions and consequently a change in the membrane potential. And depending upon the neurotransmitter, we have two types of neurotransmitters, the inhibitory and the excitatory. If the neurotransmitter is inhibitory, this will hyperpolarize the postsynaptic membrane. But if the neurotransmitter is excitatory, this will depolarize the postsynaptic membrane. We will talk about the neurotransmitters, which neurotransmitters are inhibitory and which are excitatory. Okay, and the next topic is the neuromuscular junction. I told you about synapse. So, what is a neuromuscular junction? It is a synapse between axons of motor neurons and skeletal muscle. So, the neurotransmitter that is released from the presynaptic terminal, it is acetylcholine. Remember it, that the neurotransmitter I talked about, that it is releasing from the presynaptic terminal into the synaptic cleft. It is acetylcholine, and the postsynaptic membrane, it has receptors for this, and these receptors are nicotinic receptor. And then we have the various steps of the neuromuscular transmission. And the first step is the synthesis and storage of acetylcholine in the presynaptic terminal. How it is stored? So we have an enzyme, that is the choline acetyltransferase. It catalyzes the formation of acetylcholine from acetyl coenzyme A and choline in the presynaptic terminal. So acetyl coenzyme A and choline, they form the acetylcholine in the presynaptic terminal with the help of the enzyme choline acetyltransferase. And then this acetylcholine, it is stored in the synaptic vesicles along with ATP and proteoglycan for later release. And the next step is the depolarization of the presynaptic terminal and calcium uptake. So the action potentials are conducted down the motor neuron, causing the depolarization of the nerve terminal, and this depolarization of the presynaptic terminal opens the calcium channels. Remember, in the diagram, we have discussed it, and when calcium permeability increases, calcium rushes into the presynaptic terminal down the electrochemical gradient because calcium is present in the higher concentration outside the neuron. So along the concentration gradient, it will rush into the presynaptic terminal. And because of this calcium uptake into the presynaptic terminal, there is the release of acetylcholine into the synaptic cleft. So the third step is the calcium uptake, which causes the release of acetylcholine into the synaptic cleft. What happens that the synaptic vesicles, they fuse with the plasma membrane and empty their contents into the cleft by the process known as exocytosis. And now remember that the postsynaptic membrane, it has the receptors for this neurotransmitter. So the next step is the diffusion of acetylcholine to the postsynaptic membrane, or we can call it as the muscle end plate, and then binding of acetylcholine to nicotinic receptors, because we know that the neurotransmitter that is releasing is acetylcholine and the receptors for it is the nicotinic receptors. So what happens that the nicotinic acetylcholine receptor, now this receptor is an ion channel. It is a sodium potassium ion channel, and when the acetylcholine it binds to the alpha subunit of the receptor, there is a conformational change, and that opens the central core of the channel, and this will increase the conductance to sodium and potassium. And recall that we talked about the two types of channels, the voltage gated channels and the ligand gated channels. So, this nicotinic receptor for acetylcholine at the motor end plate, this is an example of a ligand gated channel. Okay, and the next step is the generation of end plate potential in the postsynaptic membrane. Because the channel that is opened by acetylcholine, it conducts both sodium and potassium ions, but more sodium ions flow through this channel and thus creating a local positive potential inside the muscle fiber membrane that is called as the end plate potential. Okay, so the contents of one synaptic vesicle, one quantum, it will produce a miniature end plate potential that is the smallest possible end plate potential. Or we can say that it is the end plate potential that is produced by the release of acetylcholine from a single synaptic vesicle. These miniature end plate potentials they summate to produce a full-fledged end plate potential. And remember that the end plate potential it is not an action potential, but simply it is the depolarization of the specialized muscle end plate. And in turn, this end plate potential initiates an action potential that spreads across the muscle membrane and thus causes the muscle contraction. Okay, so the next step is the depolarization of the adjacent muscle membrane to threshold. Now, once the end plate region is depolarized, local currents they cause depolarization and action potentials in the adjacent muscle tissue. Action potentials in the muscle are followed by contraction. And the last step is the degradation and destruction of acetylcholine. So the end plate potential is transient because acetylcholine is degraded by an enzyme, and that is the acetylcholinesterase on the muscle end plate. Remember this enzyme. So acetylcholinesterase it will degrade the acetylcholine back into the acetyl coenzyme A and choline. And this choline, one half of the choline is taken back into the presynaptic ending by a cotransport, that is the sodium choline cotransport, and that is used to synthesize the new acetylcholine. Okay, now we have talked about the neuromuscular transmission. Now let's talk about certain pharmacological agents that will affect this neuromuscular transmission. So there's a table here in BRS Physiology. And the first example is a botulinum toxin. So, what is its action? It blocks the release of acetylcholine from the presynaptic terminals, and this causes the total blockade of neuromuscular transmission. So, botulinum toxin, it causes the blockage of neuromuscular transmission. And the next drug is the curare. So, it competes with acetylcholine for receptors on the motor end plate. So, it is kind of a competitive inhibitor for acetylcholine. It will compete with acetylcholine for receptors on the motor end plate. And what is the effect on neuromuscular transmission? This decreases the size of the end plate potential, and maximal doses, they will produce paralysis of respiratory muscles and can also cause death. And the next drug is the neostigmine. It's very important. Definitely remember this, that it inhibits the enzyme we have talked about, the enzyme acetylcholinesterase, that is causing the degradation of acetylcholine. So neostigmine inhibits that enzyme, and what happens that it will prolong and enhance the action of acetylcholine at the muscle and plate. And hemicolinium is the last drug in the table. So it blocks the reuptake of choline in the presynaptic terminal. The choline that is taken back into the presynaptic terminal by the choline and sodium co-transport. So hemicolinium, it blocks this reuptake of choline in the presynaptic terminal, and this will depletes the acetylcholine stores from the presynaptic terminal. And the last thing related to the neuromuscular transmission, we have a disease known as myasthenia gravis. So, it is an autoimmune disease that is caused by the presence of antibodies to the acetylcholine receptor. And this is characterized by skeletal muscle weakness and fatigability resulting from the reduced number of acetylcholine receptors on the muscle and plate. The size of end plate potential is reduced, and when the size of end plate potential is reduced, it is more difficult to depolarize the muscle membrane to threshold and to produce the action potentials. And how we can treat it? So, the treatment of myasthenia gravis is with acetylcholinesterase inhibitors, that is neostigmine. And this prevents the degradation of acetylcholine and prolongs the action of acetylcholine at the muscle and plate, partially compensating for the reduced number of receptors. Okay, guys, that's it for today. Keep watching my videos, like, share, and subscribe to my channel. I'll see you in the next video.